So hello everyone, thank you all for coming. First, I'd like to take the opportunity to thank ISCB and ISMB for giving me the chance to talk with you today. Like Russell mentioned, my work today is going to be on Pareto-Optimal Phylogenetic tree reconciliation, and it's actually joint work with Ron Liebitz-Hadas, Muko Bonso, who's in the audience, and also Manoli Kellis. So the problem we're going to try to consider today is if we have a set of genes from multiple species, how might we determine how they have evolved? And to understand this gene family evolution, we're going to be interested in two types of trees. So one is the species tree, so that gives us the history of a set of species. So for example, here, every time we see a bifurcation, that means that an ancestral species has split up into two non-interbreeding populations. Within the species tree, we might have a gene tree that shows us the history of the set of gene sequences. So in the simplest case, the gene tree that in black and the species tree in blue are congruent, and that means they have the same gene topology, the same topology. So in this case, every bifurcation in the gene tree is induced by a speciation in the species tree. But of course, we know it's not always this simple. So for example, we might have duplications and losses. So for example, in this case, we have a duplication in this ancestral species that's created two children lineages that continue to evolve. Also down here, we have this loss event in this leftmost species. Within prokaryotes, we might also have events such as horizontal gene transfer. So here, in contrast to vertical transmission from a parent to a child, we have transmission of genetic material between coexisting species. Another event that might cause incongruence is coalescence, in particular deep coalescence. So here, because populations are actually made up of multiple species, you could have incongruence just due to sampling effects. So the problem I'm going to consider is if we're given a species tree and a gene tree topology, for example, from your favorite phylogenetic program, how might we actually determine the reconciliation or the nesting of this gene tree inside the species tree? And that's what actually determines the evolutionary events. So for example, duplications, transfers, and losses responsible for the current gene family that we see. So I'm going to be focusing in this talk on evolution with duplication, transfer, and loss. So evolution that's mostly applicable to prokaryotes, but also to some eukaryotes. So in this case, let's consider we have the species tree shown here with an ancestral genomes X, Y, and Z. And we're going to see how a gene might evolve within the species tree. So in this case, we have this gene tree. Within species X, we have one gene, and within the time period of species X, nothing has happened. So down here at the speciation event, we still have one gene lineage. This gene lineage then speciated, so each descendant species has a copy of this gene. And here along species Y, we also have this duplication that created two children copies. If we keep evolving, in this case, along species C, we have a loss. And if we keep going, right now I've shown a transfer from species D to species C. So the reconciliation problem is, instead of going forward in time, we're given these two gene topologies, and we're also given the leaf mapping. So we know that, for example, gene A1 and A2 come from species A, B1 from species B, and so on. And what we're trying to figure out is a way of mapping this gene tree within the species tree. And the way I'm going to represent this is we're going to try to map every node in this gene tree to the species in which it evolved, and also to the event that corresponds to this bifurcation. So for example, here, this most ancestral node evolved in species X, and it corresponds to the speciation here. So if we want to try to solve this detailed reconciliation problem, there's multiple ways to do it. And the most popular approach that people use right now is using parsimony, in particular maximum parsimony. So here we are given user-specified costs to each event, so duplication, transfer, loss, or each assigned a positive cost. Usually we assume speciation is a null event, so it has zero cost. And the goal is to find the reconciliation with the minimal total cost. You can see that if we have a proposed given reconciliation, it's very easy to determine the total cost. So for example here, if we have one duplication, one transfer, and two losses, then we can just multiply by the corresponding event cost and figure out the total cost for this reconciliation. And obviously, different reconciliations can have different costs. The reconciliation problem in a DTL is very well studied. There's a lot of different variants. So for example, you could have dated or undated species trees, and time-consistent or time-inconsistent approaches. By this, I mean, because of transfers, you might induce cycles inside the gene tree. And time-consistent approaches make sure that you don't have any of these cycles where a gene is its own ancestor. There's also more recent work in trying to figure out probabilistic approaches to reconciliation, though parsimony is more well used in the community right now. There's also lots of programs and software tools along these different variants. And today we're going to consider building on an algorithm developed by Mukul Bansal, one of the authors of this paper, that was actually presented at ISMB 2 years ago. And in this paper, he proposed a big O of Mn time complexity algorithm for solving this problem, where M and N are the number of nodes in the gene and species tree. And in this variant, we're looking at an undated species tree, and the solutions are not necessarily time consistent, though they tend to be in practice. So the problem is that the optimal reconciliation that we find depends on these event costs. So for example, in this case, I have this species tree and gene tree, and I've given these user-defined event costs, so 2, each duplication costs 2, each transfer costs 4, and each loss costs 1. Then the best reconciliation I find might have 1 duplication, 1 transfer, and 2 losses. If I change my event costs, so now duplications are 1, transfers are 2, and losses are 1, then I might get a different reconciliation that's optimal. So here we might have 2 transfers. So the question is, can we actually systematically analyze the relationship between these event cost assignments and the resulting NP reconciliations? And the solution I'm going to use is, rather than trying to optimize for this total numerical cost, this total reconciliation cost, I'm going to actually optimize for the event counts. So here we're going to consider an event count vector associated with each reconciliation. And so this vector, it says how many duplications, delta, transfers, data, and losses do I have. And I'm not going to count the speciations, 1, because they are my null event, and 2, because it turns out that if we're given the number of duplications and transfers, the number of speciations is actually implicit. And here we're going to say that a vector v is strictly better than another event count vector v prime if 2 conditions are true. So each entry in v has to be less than or equal to the corresponding entry in v prime, and at least 1 entry of v has to be less than the corresponding entry in v prime. And this is important because this means that no matter what my event cost assignments that I choose, if v is strictly better than v prime, then for all possible event cost assignments, v will induce a better reconciliation cost than v prime. Finally, I'm going to say that the reconciliation is Pareto-optimal, or if there exists no reconciliation with a strictly better event count vector. There has been some previous work on looking at these Pareto-optimal reconciliations. So Charleston in 1988, and this is implemented in the software package TreeMap in Jungles, actually considered Pareto-optimal reconciliations. The problem is his algorithm has exponential time complexity, so it can actually only be used with very small trees, like 5 or 10 nodes. Tofi, in his PhD thesis, also looked at Pareto optimality. The problem with his solution is that it does not account for losses. And this simplifies the algorithmic problem, but it's been shown that not considering losses actually reduces accuracy by quite a bit. So this means what we need is actually new algorithms and tools for efficiently computing and exploring the space of Pareto optimal solutions. So the contributions that we provide in our paper is we provide a big O of m to the 5th n log m time algorithm to actually compute these Pareto-optimal event count vectors. And we have a accompanying algorithm that's m to the 4th log m time complexity to partition the event cost space into equivalence classes. And I'll talk about this when I get into the results. In conjunction with this, we're also going to provide 3 applications and software tools to actually use this algorithm in a meaningful fashion to analyze your own data sets. Like I said, what we're going to do is our algorithm is going to build on this big O of Mn time algorithm developed by Bansal. And rather than using these explicit event cost assignments, we're going to keep track of all of the event counts and Pareto-optimal solutions. So now I'm going to talk a little bit about the algorithm. For sake of time, I'm just going to give a brief sketch of the algorithm, but the full details are, of course, provided in the paper. So my dynamic programming algorithm, what I'm going to do is I'm going to consider each gene node in the gene tree in post-order traversal and each species node in the species tree. I'm going to define a set P that takes parameters G and S, and that set is going to be keeping track of the number of the set of Pareto-optimal event count vectors that reconcile this node G to the species node S. And similarly, we're going to define P speciation, P duplication, and P transfer to be the same mapping G to S, but we have the restriction that G is a speciation, a duplication, or a transfer, respectively. So So to start off my dynamic programming algorithm, what's going to happen is I'm going to consider mapping a node of a gene tree into the different nodes of the species tree. So in this case, if I'm mapping from this A1 to big A, I am mapping to the node, the species in which it was sampled. And so that's actually going to induce 0 costs. And so this is going to have 0 duplications, 0 transfers, and 0 losses. If I map it anywhere else, so if A1 goes to B, A1 is C, maps to C, or so forth, that's actually impossible. And so we're going to give it infinity event costs. And the crux of our algorithm is actually how do we consider all the other possible mappings of G to S, so the ancestral node mappings. And in this case, what we're going to consider is we're going to consider what happens if we map G to a speciation, G to a duplication, and G to a transfer. And here what I'm showing is an operation. So this is a plus operation. And what that basically does is it combines these different sets of Pareto-Optimal event count solutions. So it takes the union and then filters it back down to the Pareto-Optimal solutions. So to compute these internal nodes, what I'm going to do is go in nested post-order traversal of my 2 trees. And so in nested post-order traversal of the gene tree, consider all possible mappings to nodes in the species tree. And for each one of those, I'm going to compute these 3 different sets, P speciation, delta transfer, and then use those to compute my P of G of S. So let's consider one specific example. Here I'm going to show what happens if I map G here to this species node S. And in this case, this is a simplified example where S only has these 2 children, S prime and S double prime, that are leaves of the species tree. So here I have 2 cases for P speciation. So either G prime can map to S prime, and G double prime can map to S double prime, in which case we'll have to consider is all possible sets of Pareto-Optimal solutions for the mapping of G prime to S prime, and the mapping of G double prime to S double prime. And this cross operation is basically a way of combining these 2 different solutions. So what this cross operation does is it takes the Cartesian product, so each pair of Pareto-Optimal solutions in these 2 different sets, it adds them together, and then once it has all the different solutions, it filters back down to the Pareto-Optimal set. So that's one case where I have G prime to S prime and G double prime to S double prime. The other case I have to consider is if I map G prime to S double prime up here, and G double prime to S prime. And it's going to induce a very similar cost, just with G prime and S prime changing respectively. So now what happens is if I want to consider how P speciation is computed, all I have to do is, again, add these 2 different event count vectors, sets of event count vectors, and filter back down. And that's that plus operation. Finally, my algorithm terminates once I reach the root of the gene tree. And then once I'm there, all I have to do is consider all possible mappings to different nodes in the species tree, and then again find the Pareto-Optimal event mapping. So just a quick aside on the runtime analysis. Again, this is going to be a sketch of the runtime, but the full details are in the paper. And what we've proven is that this algorithm that we've developed has a runtime complexity of big O m to the 5th n log m. And the sketch looks something like this. So because we're doing a nested traversal of the 2 trees, that means we're going to consider m gene tree nodes and n species tree nodes. So we have a big O of mn computations as my nested loops. Within that nested loops, what I have to consider is the complexity of my different computations. So here I have a complexity of my plus operation and of my cross operation. And it turns out that the complexity of the plus operation can actually be proven to be at most big m to the 4th. The complexity of the cross operation is O of m to the 4th log m. And so all we have to do is multiply this nested loop by my most expensive operation, so this one right here, and that gives me my total time complexity. So now that we have this DP algorithm, what we're going to consider is, let's assume we have this event cost space, and what we want to do is actually partition it into disjoint regions, such that all event cost assignments within the same region induce the same set of NP reconciliations, so the same set of optimal solutions. Again, I'm going to assume that speciation is a null event of cost 0, and I'm just going to normalize duplication to cost 1, so that's without loss of generality. But what that does is allow me to give transfers with losses any positive cost with respect to the duplication, and what we have is just a 2D space of transfer and loss costs. And I'm not going to go into the details of this, but it turns out that you can actually compute these partitions in O m to the 4th log m time. And again, full details in the paper. So before I go on to the results of this of a paper, I just want to make a side that right now I've been talking about this duplication-transfer-loss model in terms of gene family evolution, but it turns out it's actually applicable across multiple contexts. So for example, we can consider the co-evolution of two different species. So here I have a gopher species tree and a louse species tree. And instead of looking at how the gene family evolves within the species tree, I'm looking at how the parasite, so the louse, evolves within the host, so the gopher species tree. And here, instead of my speciation, duplication, transfer, loss, I have co-speciation, independent speciation, host switches, and losses. This DTL model actually turns out to be applicable also to the phylogeographic domain. So here we're considering how a species tree might map within the area cladogram. So now I'm going to talk a little bit about the software tools that we've developed to make use of this algorithm. So the first one is if we want to consider the cost landscape. So we denote this by costscape. And here what we're trying to see is we want to compute the Pareto-Optimal event count vectors and the corresponding regions. So before this algorithm, what will happen is if we have the co-evolution of gophers and lice, and we have this 2D landscape of transfer costs and loss costs, then previous to a paper, what you would do is you would use an algorithm and probably the default cost. And for example, if you use angst or ranger DTL, you have this default cost and you induce this solution. If you use a different software with different default costs, you might have this solution. But what about everything else in this space? Well, now using our algorithm, we can actually fill that out quite efficiently, and we get something like this. So here what we're saying is every point in this red region is a cost assignment, and every one of those cost assignments will induce the same optimal reconciliations. So in this case, this red one actually induces 6 speciations, 0 duplications, 3 transfers, and 1 loss. And turns out there's 2 reconciliations that have the same event cost vector. Another important thing to notice from this is that now we can also find these, what we term degenerate regions. So these are, for example, lines or points. And if you consider previous to our algorithm, if you had a sample, it would be very unlikely that you might find a point along this edge. So also, in addition to this gopher and lice, the cost landscape might be more complicated. For example, here I'm showing what happens between the co-evolution of indigo birds and finches. And you can see we have many more optimal event cost vectors, and also the landscape is much more complex. So what this shows is that selecting a single set of event cost assignments might miss potentially important parts of the solution space. And now we're able to actually enumerate and show what that event cost landscape looks like. Another application we might have is if we want to look at the significance landscape, or SIGscape. So this is particularly applicable to the co-evolution world. And here what we're trying to test is the null hypothesis that the host and parasite trees are similar due to chance. So what we do is we perform k trials, and each trial we permute the leaf mappings. And then we want to compute an empirical p-value that represents the fraction of trials whose cost is less than or equal to the cost of a non-permuted data set. So if this p is less than some alpha, so some significance threshold, then we say that we reject the null hypothesis and they did co-evolve. So now using our algorithm, what we can do is actually, over this cost landscape, we can find all the regions where we do have significance. So for example, p less than 0.05. Regions where we might have significance, so p between 0.05 and 0.1, and also p where we have no significance. So if we're in this red region, we actually cannot reject the hypothesis that they are similar due to chance. Right, so the idea of this is that these conclusions of significance, so co-evolution versus non-co-evolution, actually can change depending on the event cost assignments. The final software tool we provided is trying to look at the event landscapes. So here we're going to compute the set of events common to every reconciliation in the region. And we're going to collect these events and partition them into sets of events found in just one region, just 2 regions, and so on. And the application of this, or one possible application, is if we want to identify events that are highly supported by merit of appearing in multiple regions. So for example, an event has strict consistency if it appears in all possible reconciliations across the entire landscape. So here, if we look at this example, we've looked at 5 different cases of co-evolution, and we see that speciations in blue tend to be pretty well represented, but transfers sometimes still survive, but duplications and losses actually are not very robust to my event cost assignments. And then if we look at majority consensus, we do slightly better. Speciations tend to be highly supported, and the other events somewhat supported. Right, so what this says is that many of these deferred events, in particular the duplication, transfer, and loss events that we might be interested in, do turn out to be highly specific to these event costs. So I'm just going to also mention that we have applied an algorithm to gene family evolution. Here for our data set, we looked across about 5,000 gene trees from across 100 species. This is a data set provided by Lawrence David and Eric Alm, who have talks this weekend. And here, what we've done is we've looked at 20 randomly sampled species and about 3,500 gene families from these species. And we've looked at cost ranges from 0.5 to 2. So here we look at the cost scape. What I'm plotting is the number of regions we have versus the number of genes. And you can see we can go up to very large gene families, so here about 80 genes. And as you might expect, the number of regions increases with the number of genes. But also, more importantly, the number of degenerate regions increases very dramatically. So this is, again, that the conclusion that the selecting of one single event cost assignment might not be representative. Within eventscape, we see similar results as before. So speciations are very robust, but the other events are not so much. I also want to mention that the algorithms and tools are highly efficient. So here they run on the order of seconds, and here I've shown mean and median runtime. The mean runtime sometimes is kind of high because we have these large gene families, but you can see the median runtime is on the order of seconds again. So finally, in summary, what we've done here is we've presented the first polynomial time, so official algorithm for computing these Pareto-optimal reconciliations. And that makes it now tractable to systematically understand the relationship between these event cost assignments and the NP reconciliations. I've shown how this is applicable to the phylogenomic and the co-phylogenetic communities, but again, it's also applicable to biogeographic analysis. And in my results, I've shown that some of these events might not be robust, and we might actually want to explore these space. And so this motivates further investigation into the robustness of previous analyses based on detailed reconciliation. Finally, the tools and the source code is available for you guys to use and download and modify as you see fit. So finally, I would like to acknowledge my collaborators in this project. So Ron Liebeskein-Hadas from Harvey Mudd, Muko, who's actually sitting here in the audience, and also my advisor, Manolo Kellis. I would also like to thank our funding agencies, and also the MIT Computational Biology group. And I would like to encourage all of you to look at some of the posters from our group listed here. I'd now be happy to take any questions.
Hey, I was wondering how your method can handle errors in the input trees.
Mm-hmm. That's a great question. So in this case, what we're assuming is that the gene tree and species tree are given to us by a phylogenetic program. And so we're not considering errors, but we have definitely looked at cases where we might modify the species tree or gene tree and account for topological errors due to like uncertainty. And so that's definitely an active area that we're investigating. It's not incorporated into the current algorithm, but right now we're combining them.
I'm wondering, how often do you find you have more than one solution?
Mm-hmm. So if we go back, right, so for example here, then actually one solution is just right way down here. But most of these gene families, in this case, I believe it was about 86% of the gene families actually had more than one event cost vector that was optimal.
Do you have like an expected or average number of solutions?
So in this case, most of them were below 5 solutions, but there were definitely some that were quite high, especially if you consider these non-degenerate regions. And then these solutions, again, they could be quite different just based on the different assignments.
A second minor question is, as for the input he mentioned, can you accept non-binary trees?
Right now, it's binary reconciliation, but again, non-binary reconciliation is something we're trying to expand into.
Thank you.
Sure. Sure.
So I'm curious, if you are trying to reconcile many gene trees with the same species tree, can you save anything beyond just running the algorithm many times? Is there any room to optimize?
Right, so right now what we're considering is, I guess, each gene tree to each species tree kind of individually. but there's definitely room for trying to kind of do all the gene trees simultaneously within the species tree and seeing if the information from the multiple gene trees can kind of force you into a particular event region.